Hello friends, welcome to my channel Botanical Studies. In this video, we will discuss about plant-water relationship, part 3rd, that is transpiration. The discussion will go through following points: definition and types of transpiration, starch-sugar interconversion hypothesis, significance of transpiration, and finally factors affecting on transpiration. Let us discuss the definition and types of transpiration. Water absorbed by the root system from the soil is transported to aerial organs through xylem. In aerial organs, very little amount of water is retained and utilized for metabolic reactions, and remaining amount of water is lost in vapor form in the surrounding atmosphere. The loss of water from aerial organs in the form of water vapor is called as transpiration. In plant-water relationship, transpiration is the final process on the basis of passage through which plant gives out water in the form of vapor. Transpiration is of three types: cuticular transpiration, stomatal transpiration, and lenticular transpiration. Let us discuss the first, leaf anatomy. A transverse section of typical leaf shows that the leaf is covered by epidermis on both surfaces, the upper epidermis on the upper surface and lower epidermis on the lower surface. The top layer is a waxy cuticle which prevents water from being absorbed. Upper epidermis, a protective layer of cells that produces the cuticle. The epidermis is also transparent and very thin to allow maximum light penetration. Palisade Misophy, a rod-shaped cells that contains large number of chloroplasts for photosynthesis. They are upright, elongated, and tightly packed together in order to increase the surface area for light absorption. Spongy Mesophy. These cells are smaller than Palisade Mesophy and are found in lower part of the leaf. Spongy mesophyll cells stores the food material. Vein. The veins consist of xylem, that is vessels that carry water, and phloem, vessels that carry dissolved nutrients such as sugar. These vessels play an important role in transporting water to chloroplasts in the mesophyll tissue for photosynthesis. They also transport the sugar produced by photosynthesis away from these cells to the rest of the plant tissue to be used as the energy source or stored. Lower epidermis, a protective layer of cells. The lower epidermis produces a waxy cuticle in some plant species. The lower epidermis contains small pores called stomata that allows carbon dioxide and oxygen to move in and out of the plant respectively. And these stomata are also helpful in the process of transpiration. Let us move towards the types of transpiration. Cuticular and stomatal transpiration. Cuticular transpiration. The loss of water through cuticle, that is a layer of waxy substance present on the surface of epidermis of the leaves, is called cuticular transpiration. This is the day-night process in which plants gives out 5 to 10% of water in the form of vapor. Stomatal transpiration. Stomata are minute pores present in the epidermis of aerial organs of a plant. They are surrounded by specialized epidermal cells known as guard cells. The loss of water through stomata is called as stomatal transpiration. The stomatal transpiration takes place when stomata is open, and it does not take place when stomata is closed. In this method, plants given out 80 to 90% of water in the form of vapor. Mechanism of stomatal transpiration. It shows two steps: diffusion of water from moist cell wall of turgid mesophyll cells into intercellular spaces and substomatal spaces, and second, the diffusion of water in vapor form from the substomatal cavity into the surrounding atmosphere when the stomata is open. The opening and closing of stomatal pore is due to movement exhibited by guard cells. In dicots, the guard cells are kidney-shaped, while in monocots they are dumbbell-shaped. The inner wall of guard cell is thick, while the outer wall is thin. The difference in thickness of the wall is responsible for movement in the guard cells. When stomata are open, the guard cells are turgid, and when stomata are closed, the guard cells are flaccid. When guard cells absorb water, their turgidity increases, which increases the pressure on the wall. Due to increase in the wall pressure in outward direction, the outer thin wall bends and becomes convex. Such bending of outer thin wall pulls the inner thick wall in the outward direction. This pulling is responsible for movement of two inner thick walls of the two guard cells away from each other. When two inner walls of the guard cells moves away from each other, the space surrounding the pore widens and the stomata opens. When the guard cell lose water, the inward pressure is created by the outer walls on the inner thick walls, due to which the two inner thick walls of the guard cells come close to each other. When two inner walls becomes close to each other, the stomatal pore closes or stoma closes. This indicates that increase in the turgidity of the guard cell is responsible for opening of the stomata, and decrease in the turgidity is responsible for closing of stomata. Starch sugar interconversion hypothesis. To explain the cause of turgidity change in guard cells, the two important theories are proposed. First, photosynthetic theory, and second, starch sugar interconversion hypothesis. Starch sugar interconversion hypothesis has been proposed to explain the mechanism of stomatal transpiration. Most of the transpiration takes place through stomata. Stomata are usually concerned in transpiration as its guard cells exhibits closing and opening movements. Closing and opening of stomata, which explains by sugar hypothesis. It explained as follows. In light, that is during daytime, photosynthesis takes place, which decreases carbon dioxide concentration, as a result of which there is an increase in the pH. Due to increase in the pH, there is a conversion of starch into sugar, because of which there is an increase in the cell sap concentration and endoosmosis of water takes place, which increases the turgor pressure and due to which the stomata opens. And in the dark, that is in the night, there is no photosynthesis, there is an increase in the CO2 concentration which decreases the pH and conversion of sugar to starch, because of which there is a decrease in the cell sap concentration and exoosmosis of water takes place, which decreases the turgor pressure and the stomata closes. Significance of transpiration. There are two views which says that the transpiration is a useful process or it is not a useful process. According to the first view, transpiration is a useful process. During upward movement of water, the essential action force developed due to transpiration. The diffusion of water and minerals from the soil into the plant body and from one cell to another in the plant body is due to suction force in the form of transpiration pool in the central conducting strand. Transpiration is responsible for removal of excess amount of water from plant tissue. Transpiration is responsible for maintaining internal temperature constant in the leaves, and it avoids the effect of excessive heat in the aerial organs exposed to sunlight rays. Opening and closing of stomata during transpiration is helpful for the gaseous exchange during photosynthesis and respiration. Transpiration is useful in translocation of food material. Transpiration helps in rapid transport of essential minerals absorbed by the root system to aerial organs. According to the second view, transpiration is harmful process, not essential for plants. Transpiration does not help in the upward movement of water. Diffusion of water and minerals from soil to root and in the plant organs occur even in absence of transpiration. Many xerophytes in which transpiration is nil or very low. Transpiration does not play any role in mineral absorption. In absence of transpiration, mineral absorption takes place and it is an independent active process. Transpiration is not responsible for removing excess amount of water from plant tissue. Transpiration is not responsible for maintaining internal temperature constant in aerial organs. Factors affecting on transpiration. There are both external as well as internal factors affecting on the rate of transpiration. The external factors are atmospheric humidity, atmospheric temperature, wind velocity, light, water present in the soil, and atmospheric pressure. Let us discuss one by one. The external factors affecting on transpiration. First, atmospheric humidity. Humidity is the actual water content of air and it is expressed in terms of relative humidity. During transpiration, water vapor diffuses through stomata in the surrounding atmosphere. The rate of this diffusion depends upon the concentration of water in the atmosphere and the concentration of water in the substomatal cavity. When the atmospheric humidity is more than the humidity in the substomatal cavity, the rate of diffusion of water in the vapor form from the plant body is very low and transpiration is low. When atmospheric humidity is less, the rate of diffusion of water from substomatal cavity is more and transpiration occurs at higher speed. For this reason, during summer season, when atmosphere is dry, the transpiration rate is high, and during rainy season, when atmosphere is humid, transpiration is very low. Atmospheric temperature. The effect of atmospheric temperature on the rate of transpiration is indirect. Temperature affects the humidity. During summer, high temperature reduces the atmospheric humidity and makes the atmosphere dry. During rainy and winter season, atmospheric temperature is low and therefore atmosphere remains more humid. Therefore, when atmospheric temperature is high, rate of transpiration is more, and when atmospheric temperature is less, transpiration is also less. Wind velocity. Wind affects the transpiration by removing the humid atmosphere near the surface of the leaves. That is, due to wind blowing, atmosphere becomes dry near the leaf surface. For this reason, when wind blows, the moderate velocity, that is, the rate of transpiration increases. When wind blows with high wind velocity, initially transpiration rate increases. And to check the transpiration, when stomatal closure occurs, the rate of transpiration decreases. Light. Like temperature, light factor affects the process of transpiration indirectly. When light intensity is moderate, rate of transpiration is normal. When the light intensity increases to high or very high light, that is during noon in summer season, stomata are closed. This high or very high light intensity are responsible for increase in the atmospheric temperature. At high temperature, to check the rate of transpiration, stomatal closure occurs. When stomata are closed at high light intensity, rate of transpiration decreases. Water present in the soil. Soil is water supplying system for the plant and water is present in the soil. It is absorbed and supplied the aerial organs. Such supply of water is essential to compensate loss of water by transpiration. When amount of water in the soil decreases, the rate of absorption and the rate of supply of water also decreases. Under such conditions, to check the water loss, rate of transpiration also decreases. Atmospheric pressure. At higher altitude, atmospheric pressure also affects the rate of transpiration. When there is reduction in atmospheric pressure, the atmospheric density reduces and it affects the process of diffusion of water at low atmospheric density. Diffusion of water is with high rate, therefore at high altitude in hilly regions, transpiration rate is high. To check the transpiration, the plant growing in such regions develops xerophytic characters like thick cuticle, sunken stomata, etc. Internal factors affecting on transpiration. The internal factors are stomatal size and density, anatomical characters in leaf, root shoot ratio. Stomatal size and density. The number of stomata per unit area square centimeter is called stomatal density or stomatal frequency. The plants with larger stomata and with high frequency or density exhibits higher rate of transpiration. Due to smaller size and less number per unit area, xerophyte shows relatively less transpiration and slow rate of transpiration. In mesophytes, higher stomatal density and larger stomata are responsible for relatively higher rate of transpiration. Anatomical characters in leaf. Leaf is the main organ for transpiration. The leaf anatomical characters are as per the amount of water available in the habitat. The plants growing under xeric conditions shows adaptation for economy of water and to check the loss of water. Plants growing near water generally do not show any characters to check transpiration. The plants with reduced release with thick cuticle and sunken stomata shows relatively less transpiration and slow rate of transpiration. Root shoot ratio. Root act as a water absorbing system while shoot act as a water transpiring system. Generally, the total surface area of water absorbing system and total area of transpiring organs controls the rate of transpiration in the plant. When area of absorbing organ is more than that of transpiring organ, the rate of transpiration is high. When the area of transpiring organ is more than the absorbing organ area, transpiration is at slow rate. In herbaceous plants, for example grasses, are with larger absorbing surface area, exhibits more water loss by transpiration than the trees with relatively larger area of transpiring surface than their absorbing surface area.